Hello students, so in this video I'll be talking about meiosis in details. In my earlier video I discussed about cell cycle and mitosis and what is the significance of mitosis. So what is basically meiosis? Meiosis is called reduction cell division. Now why it is called reduction cell division? Because here the chromosome number gets or becomes halved. So if you begin with a cell which is diploid, you will end up getting two cells which are having half the number of chromosomes. That's why it is called reduction cell division. Now just like you have got four steps in case of mitosis: prophase, metaphase, anaphase and telophase. Similar are the steps of meiosis, but here there are some extra steps. So meiosis, when we are talking about meiosis is basically occurs in two steps. One is meiosis 1. Meiosis 1 is also called the reduction phase. Now why it is called the reduction phase? Because it is during this phase that the number of chromosomes will become half. And meiosis 2, it is called the mitotic phase. That means followed by meiosis 1 you will have meiosis 2, and meiosis 2 is very similar to mitosis. Okay, now why meiosis 2 is required, that we'll be discussing later in this video. So first let us look at the names of the different steps. So you have the four essential steps of karyokinesis: prophase, metaphase, anaphase, telophase. So if you remember the first letters of the steps like P, M, A, T, so it forms PMAT. So like that you can remember: prophase, metaphase, anaphase and telophase. So here prophase 1. Prophase 1 is quite lengthy because it has many substages which are leptotin, zygotin, pectine, diplotin and dikinesis. Then we have metaphase 1, anaphase 1, telophase 1. Meiosis, which will be followed by, of course, cytokinesis 1. These are all karyokinesis 1. That means the nuclear division, which will be followed by cytokinesis 1. After cytokinesis 1, meiosis 2 will begin, where you will have prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis 2. All these steps are very, or you can say exactly same like that of mitosis. So I have written significance of meiosis here, but that I will discuss later on in the video. First let us look at what happens during these stages. So I have drawn in an elongated way. We'll proceed. So you know that in the I phase or the interphase, what happens? That the nuclear membrane is intact. The chromosomes are in the form of chromatin fibers or chromatin network. There's the centrosome having two centrioles. There's the animal cell. Plant cells don't have centrosome. Now, as from I phase, the cell will enter into prophase 1. Now, prophase 1 had different substages. So, first is leptotin. So, during all these stages of prophase 1, I have written prophase 1. During all these stages of prophase 1, you will find that what will happen? The nuclear membrane will slowly keep on disappearing. At the end of prophase 1, nuclear membrane will totally disappear. Spindle fiber formation will be complete. So, those stages I have not shown properly here because those will be happening in the background. We are more concerned about what will happen to the chromosomes. So first is leptotin. During leptotin, what happens? So just like I explained in mitosis, here also I have taken one pair of homologous chromosome indicated by black color and red color. So there is a pair of homologous chromosome. So there is a diploid cell. Slowly the chromosomes will start condensing. They will become thick rod-like structure. From leptotin, as we proceed into zygotin, what will happen? These chromosomes, suppose this is one chromosome, this chromosome will split from center longitudinally and it will end up forming two chromatids, and they are joined in the center, and this point you know is called the centromere. So that will happen during zygotin. After that, a very interesting and a very important step occurs, which is called pectine. So what happens during the pectine stage? If you can see here, I'll just focus. Okay. So if you can see here, so each chromosome, they split longitudinally into chromatids. So these two are sister chromatids, these two are sister chromatids, but this one and this one are non-sister chromatids. So during the pectine stage, there will be crisscross of the non-sister chromatids at different points. I have shown just 2 points of crisscrossing, but at multiple points crisscrossing takes place. And this process is called crossing over. Crossing over is a very important phenomenon because as they will crisscross, they will exchange their DNA segments. That means if you can see here properly, this black color part will get cut here and will get attached with this red color portion, and this red color portion, it will get cut here and it will get attached with this black color portion. So there is a shuffling of the genes between the different chromosomes, and this point of crossover where they form this crisscross, this point is called the chiasma, chiasma, chiasma. So that is what happens during pectiting. Crossing over takes place during pectiting. It has a very big significance. It causes variation. I'll be discussing that in the significance of meiosis. After that, we proceed to the next step, which is called your diplotin. So during diplotin, already crossover has taken place. So you can see here that this black color part has got joined with the red color part, and here this black color part has got joined with this red color part of the chromatid. So they will separate from each other after exchanging the chromosomal segments, but they will remain attached at the point of crossover in diplotin. And dikinesis, kinesis you know is separation. So during dikinesis, now both the portions of crossover, they will totally get separated from each other with the crossed over segment. So it is very clear from here, this black color part has got attached with this red color portion, and this red color portion has got attached with this black color part. So exchange of chromosomal segments have already taken place, and separation occurs during dikinesis. They have got separated out, and in the background, if you can see faint lines I have drawn, so spindle fiber formation also gets completed. So all these stages, leptotin, zygotin, pectitin, diplotin and dikinesis, together it forms the prophase 1. After that, the things will proceed normally, this will be followed with metaphase 1. So you know what happens during metaphase. During metaphase, all the chromosomes, they get aligned along the equatorial plane, along the metaphasic plane. And one more interesting thing, and the chromosomes will get attached to the spindle fibers. So after getting attached to the spindle fibers, next, so that happens during metaphase 1. Next comes anaphase. Now this anaphase is slightly different from that of mitosis. If you recall, during mitosis, what happened? The chromatids got separated and they moved in two different directions. Two chromatids, they moved in two different directions. But in case of anaphase 1 of meiosis, the whole chromosome with both the chromatids, these are two chromatids of this chromosome, and these two are the chromatids of this chromosome. So both the chromatids will move together. They will not get separated. So both the chromatids will move towards one pole. Here, both the chromatids will move towards one pole. That means the entire chromosome is coming to this pole. Here, this entire chromosome is going to the other pole. So that is what happens in anaphase 1. And that is the difference between mitosis and meiosis. Next, it comes to telophase. During telophase, what happens? The nuclear membrane will start reappearing, and this is the chromosome which reaches to that pole with the crossed over segments. You can see here also this chromosome which reached to this pole. This was the red color chromosome with black color tips, the exchanged portions. I'll try to bring it near. Okay, so I hope it is clear now. So that way, so nuclear membrane will start reappearing, and slowly it will be followed by cytokinesis 1, where the cells will divide. This cell will divide to form two cells. Now if you see, these two are the chromatids, and two chromatids, they are belonging to one chromosome. So we began with two chromosomes in one cell, and we ended up with one chromosome in one cell. So the number of chromosome has become half. That's why it is called the reduction phase. Meiosis 1 is called the reduction phase. Number of chromosomes becomes half. Next, the cell proceeds to meiosis 2. Meiosis 2 is called the mitotic phase because the steps are exactly similar to that of mitosis. During prophase 2, what will happen? Again, the nuclear membrane will start disappearing. Spindle fiber formation will start. as the cell will proceed. So two cells were formed. So I am showing both the cells how it is proceeding. From one cell two cells were formed, and from here how it is proceeding I am showing here. So what happens here in the metaphase 2, again the chromosome will get attached to the spindle fiber along the equatorial plane. Here also it gets attached to a spindle fiber, and now if you see, as I have shown there, that two chromatids get separated during mitosis, here also the same thing happens. Now at the centromer, both the chromatids will get detached. One chromatid will go towards one pole, this chromatid will come towards another pole. The moment the chromatid gets separated, they are regarded as, or they are called chromosome. I told you that in the earlier video. So here also both the chromatids are moving towards two poles. So that happens during anaphase 2. During telophase, once again the nuclear membrane will start reappearing, spindle fiber will start disappearing, and the chromosomes, now they are called chromosomes, the chromosomes will start losing their rod-like structure. They will become more fiber-like, and which will be followed by cytokinesis 2. So after telophase 2, we have cytokinesis 2. The cell will divide. This will divide to form two cells. This will divide to form two cells with one chromosome each. So what we find that in case of meiosis, we begin with one cell, okay, and we end up with 4 daughter cells having half the number of chromosomes. Now the question is, if the number of chromosomes became half during your meiosis 1, then what is the requirement of meiosis 2? To understand that, let us proceed to another section where I will be showing you how the chromosomes and DNA actually gets distributed. So there's the diagram. Okay, so just like I explained for mitosis in my earlier video, here also I'll be showing distribution of chromosome and DNA in meiosis. So I have taken here one pair of homologous chromosome. So it is a diploid condition. So twice n. DNA being an organic molecule, it is represented by C. So DNA is also in a diploid state, so it is twice C. So as the cell will pass through the S phase, number of DNA will get doubled. Okay, so amount of DNA gets doubled. So chromosome remains in a diploid condition twice n, but DNA becomes 4C. From here, I told you that the chromosomes will split longitudinally into sister chromatids. Okay, this splits into two, this one splits into two. Each chromatid will have one DNA molecule. This condition is still twice n 4C because there is one chromosome, there is one chromosome, so diploid, and 4C, that is your 4 number of DNA molecules. Because this n represents chromosomes, it's not the chromatid, that's why we are writing twice n, diploid state. So what happened in meiosis 1? The entire chromosome moved over to one cell after crossing over. You can see I have shown some regions of crossing over. So in between crossing over and everything is taking place. So after that, this whole chromosome will come to one cell, this whole chromosome will come to one cell, not like mitosis. In mitosis, what happened? One chromatid came to this cell, from here also one chromatid came to this cell. Okay, but here this entire chromosome will move over to one cell, this entire chromosome with both the chromatids will move over to one cell. So this, though there are two chromatids, but this is how many chromosome? One chromosome. That's why the chromosome number has become haploid, n. Here also one chromosome comes this side, so n. So chromosome number has become half. That's why we call it reduction phase. But if you look at the DNA amount still remains twice C. So under this condition, because meiosis leads to gamete formation, so during gamete formation, meiosis takes place. Chromosome number needs to become half. Why? To maintain the species identity. I'll explain that to you. So if the DNA amount remains double, twice C, suppose there's a sperm and there's an egg and both fuses, chromosome number will be restored in the zygote twice n, n plus n, but DNA amount will become double of the original amount. So that will no longer remain the particular species. The species quality will totally change, or properties will totally change. So this is the reason why the cells have to go through another phase, and that is meiosis 2. So in meiosis 2, what happens? The chromatids get separated. So this chromatid will come over to one cell, this chromatid will come over to one cell, this chromatid will come over to one cell, this chromatid will come over to one cell. Now I told you before also that the moment the chromatids get separated, they are regarded as chromosomes. So this is one chromosome, so N, this is one chromosome, so N, and each chromosome is having one DNA molecule. That's why the number of, or the amount of DNA has become C. So we began with one cell having twice and twice C. We ended up with 4 cells having NC configuration. Now suppose this is a sperm and this is the egg, and both fuses to form the zygote. So N plus N, it will give us twice N. Original number of chromosome is restored. C plus C will give us twice C. So the DNA amount also gets restored. So that is the significance of meiosis 2. It helps in restoring, or it helps in reducing the DNA amount. Chromosome number will get reduced in meiosis 1. DNA amount will further reduce in meiosis 2, so that when fusion of gametes takes place, the original amount of DNA can also be restored. So what is the significance? I had written before. So let's look at the significance of meiosis. So what basically happens in meiosis? Meiosis leads to formation of gametes, or during gamete formation meiosis takes place, you can say that. So number of, in the gametes, the number of chromosome needs to become half, because chromosome number present in your cell is your species identity. Suppose you get some cell somewhere, and after analysis you come to know that the cell is having 46 chromosome. So you'll come to know that this cell belongs to human being. So that is the importance of the chromosome number. Chromosome number is representative of the species identity. Now in the gamete, suppose there is an egg and this is being fertilized by a sperm, and for sperm is also having 46 diploid number of chromosome, egg is also having 46 number of chromosome, both fuses. So total number of chromosomes will become 92. So the entire species identity will change. It will no longer remain a human being. So in order to maintain the species identity, when sperm or gametes will be formed, so this chromosome number needs to become half. It needs to become 23. This also, I am giving the example of human beings, this also needs to become 23. So that when both the gametes will fuse, 23 plus 23, original number 46 number of chromosomes will be restored in the zygote, and the species identity will be maintained. So that is the very important significance of meiosis. Apart from that, I told you this also before, that crossing over occurs during meiosis. Which stage it was? It was during Pectian. And due to this crossing over, there is shuffling, just like we shuffle the cards to get new combination every time. So shuffling of the chromosomal segments take place. Crossing over of chromosomal segment means it is actually crossing over of DNA segments takes place, which leads to variation. So every time there is crossing over, a new combination comes in and we get a variety. And when there is variety, you know that it leads to better survival of a species. So this is the significance of meiosis. This meiosis occurs only in reproductive cells. Thank you so much. I hope the concepts are clear to you.